Hello everyone, welcome to all. Today we are going to handle with the topic on sexual reproduction in flowering plants. They are also called as angiospermic plants. Pistil, or it's also known as gynoecium. It represents the female reproductive part of the flower. Based on the number of a pistil, the carpels are classified into four types. First one will be a monocarpellary, second is bicarpellary, third is tricarpellary, fourth is a multicarpellary. In most of the flowers, the carpels are multicarpellary in nature. In that, some of them are apocarpous in nature and few of them are syncarpous in nature. Apocarpous means the carpels are free in nature, syncarpous means the carpels are fused in nature. And pistillar carpel, they are consisting of three major parts. They are style, stigma, and ovary. And they are also called as a female gametophyte or a gynoecium. Types of a gynoecium. First one is apocarpus. Example for it, a Michaelia and Crassula. So usually in apocarpus, the placentation pattern is a basal placentation. So in most of our apocarpus, the carpels are multicarpellary in nature and they are free in nature. Second one, syncarpus gynoecium. Syncarpus means carpels are fused in nature. Example for it, mustard and tomato. Here we can see the two kinds of a placentation. One will be axial placentation, another is a parietal or a peripheral placentation. In this picture, you can see easily the apocarpus ovary and syncarpus ovary. Apocarpus ovary is consisting of only a basal placentation, but when it comes to a syncarpus ovary, there are two kinds of placentation will be there. So one will be a axial, another will be a parietal or peripheral placentation. Inside the ovary is the ovarian cavity is present. Ovarian cavity is nothing but a locule in which the placenta is located. With the help of that placenta only, the ovules are attached inside the locule. Arising from the placenta or the ovules, they are nothing but a microsporangia. The number of ovules in an ovary may be one. Example, wheat, paddy, mango, and too many ovules might be there. Example for that, papaya, watermelon, and orchids. Structure of megasporangium. It's also known as ovule. There are several parts are there. They are as follows: hilum, funicle, micropyle, integument, nucellus, embryo sac, and chalazal pore. The first one, hilum. It is the attachment portion of a funicle and ovule. Funicle, stalk of a ovule. Micropyle, it is the opening of a ovule or a embryo sac. Integuments, it is a protective layer present on the surface of a ovule. There are two integuments are there. One will be a outer integument, another will be a inner integument. Nucellus. Nucellus is a nutritive tissue. It helps in the development of embryo sac. Embryo sac, it is also known as a female gametophyte. Chalazal pole, it is the opposite pole present to the opposite to the micropole. Just to revise these points once again, the structure of ovules and their parts and their functions. Megasporogenesis. The first point, it is the formation of a megaspore from the megaspore mother cells, that is MMCs. Second one, ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is a large cell contain dense cytoplasm and prominent nucleus. Third point, the MMCS undergoes a mitotic division. It results in the production of 4 megaspores. In a majority of flowering plants, one of the megaspore is functional while the other three degenerate. The functional megaspore develops into the female gametophyte. This method of embryo sac formation from a single megaspore is termed monosporic development. You can see the development of megasporogenesis in this picture. Initially, the megaspore mother cell divides into two equal halves to form a megaspore dyad. After that, it forms into megaspore tetrads. In that tetrads, three of them degenerate and one will become a functional. That functional megaspore forms into a embryo sac. That is nothing but a female gametophyte or a megasporogenesis formation. Formation of the embryo sac. The nucleus of the functional megaspore divides mitotically to form two nuclei. They move to the opposite poles, forming two nucleate embryo sac. The nuclei again divide two times, forming 4 nucleate and 8 nucleate stages of the embryo sac. These divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by the cell wall formation. After the 8 nucleate stage, cell wall are let down, leading to the organization of the typical female gametophyte or embryo sac. 6 of the 8 nuclei are surrounded by cell wall and organized into cells. Remaining 2 nuclei, polar nuclei, are situated below the egg apparatus in the large central cell. Out of 6 cells, 3 group of cells formed at the chalazal region and forms into antipodal cells, and 3 group of cells formed at the micropylar, and in that 2 cells are synergids and 1 will be egg apparatus. Here in this picture you can see the distribution of the cells within the embryo sac. A typical mature embryo sac is 8 nucleate and 7 celled. 3 cells are grouped at the micropylar end and forms egg apparatus. It consists of 2 synergids and 1 egg cell. Synergids have special cellular thickenings at the micropylar tip called filiform apparatus. It helps to guide the pollen tube into the synergid. 3 cells at the chalazal end are called the antipodals. The large central cell has the 2 polar nuclei. Again, back to conclusion. That is a summary of today's session. Today we discussed about a gynoecium, that is a pistil. In that, the types of a pistils, that is a mono, bi, tri and multicarpellaris, and the structure of a microsporogenesis, and the development of a female gametophyte is nothing but a megasporogenesis, and last one is the formation of embryo sac, that is 8-celled stage.